Okay, hi there. So we're going to talk about an introduction to ecology, which is chapter 1 in your textbook. And this chapter really focuses on a case study of amphibian decline. Let me try to put my picture over there so it doesn't cover anything up. So amphibians are salamanders, frogs, which are called anurans, and caecilians, which look like legless snakes, they look like snakes, but they're really legless salamanders. And global amphibian declines are an exceedingly complex ecological issue, one that really highlights the interconnectedness of ecology well. So frogs and other amphibians are really sensitive to environmental conditions. Why? Maybe pause the video and write down a few reasons that you think frogs and amphibians might be really susceptible to environmental conditions. Okay, great. So now that you have some ideas about why, let's just think about amphibians for a minute. So they're these semi-aquatic organisms. They go between terrestrial and aquatic environments. They're very susceptible to desiccation. They breathe through their skin, so their skin is very permeable, and toxins can pass through their skin as well. Their eggs are also very soft and can dry out. They don't have protective shells on their eggs, and so just like their skin, their eggs are very vulnerable to both drying out and to toxins. And because they spend time in both terrestrial and aquatic environments, they cross those ecosystem boundaries, and so they're exposed to pollutants in both places and to extinction pressures in both environments. Both aluminum, well, all kinds of things are toxic, but in particular for amphibians, things like aluminum, cadmium, and copper are all toxic elements. And those are found in quite high abundances in some human environments. So if we think about historic extinction rates for amphibians, prior to about 1848, which is around the gold rush, there were really no measurable declines of frogs. There were kind of just base extinction levels for all organisms at that time. But around 1896, when bullfrogs were introduced to California, we see this sharp increase in extinctions. And so a lot of people thought that bullfrogs were one of the main culprits. But now we know that it's actually a whole host of interacting factors. And our current extinction rates for amphibians are actually 27 times higher than former or kind of pre-European settlement extinction rates. So this is a huge cause for concern for frogs worldwide. And if you look at extinction numbers, you can see that, in the United States and North America, we have 41 extinct or critically endangered amphibians, with 107 species in threatened or vulnerable status. And those numbers are much higher in the tropics in Central America, in South America, and in parts of Indonesia and Australia and Africa. So these are worldwide phenomena for amphibians, and we'll talk about some of the global factors that are influencing extinctions. So around the 1950s, amphibians just started disappearing, and people were trying to understand why. So a number of factors have been implicated. First is just wetland loss. So wetlands, of course, are main frog habitat, and we have been draining and filling wetlands for the last 150 years, if not longer. And according to sources, which haven't been updated recently, which is really worrisome to me, but about 50% of our wetlands in the US have been lost since 1600, but these are numbers from the 1980s, and there are no recent numbers. I think if you were to look at a map from the 1980s to the 2020s, you would see wetlands being lost, but then wetlands being created. And so we're in the past 4 decades of what's called wetland mitigation, where we're trying to build wetlands in more convenient locations, but those wetlands don't usually have the same functionality that natural wetlands have. So we do have a great opportunity. The map at the bottom here shows possible restoration potential for wetlands, and so there's lots of great possibilities for restoring wetlands. whether this work is actually being done or not, that's a big question. And can you restore function completely to a wetland that's been damaged? That's another good question. So habitat destruction. Along with habitat destruction is logging. Of course, these amphibians are spending time in terrestrial habitats too, and so when you log riparian forests and you log other forests, especially in the tropics, then you have massive problems for amphibians. Along with the issues of wetlands, we also have flow alteration, dams, floodplain destruction, and the building of levees along rivers. So we know that only 2% of our rivers in North America are undammed, and this is a further cause for amphibian declines, riverine habitat destruction. Here's just a picture of the number of dams worldwide as of 2003. Of course, each of these spots is large enough to cover a huge area, and so it just goes to show how prevalent dams are worldwide. The final factor in terms of habitat destruction is urbanization, the growth of impervious surfaces like roads and parking lots, and agriculture, which has also destroyed some critical amphibian habitat. And along with habitat destruction comes really the larger issue of how might climate changes, global warming's effects on water temperature, increased storm severity, increased drought severity, how might all of these kind of untested climate change impacts influence frogs. So there are some estimates that climate change will severely impact 24 to 44% of all amphibians. Some other factors that influence amphibians in particular are things like UV radiation, because they're so sensitive and their skin is so fragile. They are very susceptible to UV radiation, which can also influence their eggs, which tend to float on the surface of ponds. And in particular, higher elevation species are very prone to these issues. So here you see some snow frogs in France. One of the most devastating effects on amphibians is the Chytrid fungus, which is called BD, Batrachotridium dendrobatus, and it's a motile fungus. So it has motile zoospores with flagella that can swim through the water and find frogs. They then penetrate the skin and grow in the skin and can cause mortality within 2 weeks. So this particular pathogen has been having devastating effects on frog populations worldwide. Another group of pathogens are called the Ranaviruses. They're a family of viruses that particularly affect fish and amphibians and other reptiles. And then there's just what are called malformations, which can be caused by a number of factors. So in 1995, students in Minnesota found large numbers of deformed frogs in a human-made wetland. They had multiple legs, they were missing legs in some cases. And by the end of 1996, there were numerous cases from all around Minnesota. And these malformations can have several causes. So one is a trematode fluke called Ribeiroia ondatrae, and it has an initial snail host that then infects frogs. And here you can see this frog with several legs. They're quite, they're creepy looking. And they're not very functional, right? So it's hard to jump around when you have more than 4 legs. Here you can see a heron is eating the frog. The heron is a host for this trematode fluke. And so the parasite lives in the heron. It lays its eggs in the water, and then they're infected by, or snails get infected. And from the snails, there's another secondary host in the amphibian. And so you can see this complicated life cycle, but it leads to these cysts that develop in the frog and cause extra limb formation. There's a controlled experiment using this system. And the way that we will talk about this, our experimental groups are compared with a control group. that lacks the factor being tested. So in this case, tree frog eggs were exposed to this Ribeiroia, fluke, we'll just call it a fluke, the fluke parasite in the lab. There were 4 treatments, 0 flukes, 16 flukes, 32 flukes, and 48 flukes. And what you can see is that the number of parasites per tadpole, as the number of parasites per tadpole on the x-axis increases, you get a percentage, you get an increasing number of frogs with deformities and a decreasing number of tadpoles surviving to metamorphosis. So in this case, the control treatment is where? So pause the video for a minute and look at the figure closely and try to figure out where the control treatment is found. Okay, great. So hopefully you figured out that the control treatment would be at 0 parasites per tadpole. And for controls, they would have 0 deformities and 85% survivorship. For the frogs with parasites, you see an increase up to like 75% of the frogs with 16 parasites had deformities, and their survivorship percentages had dropped a little bit. And then you see that only increase and decrease with increasing number of parasites. Okay, so take a quick 10-minute break. So far we've talked about a lot of things that have influenced frogs.